Immunology Part 7. Here we discuss the line of defense. Line of defense is of three types. First line of defense, second line of defense, third line of defense. Actually, in the third line of defense, discussed about the humoral and cell-mediated immunity, which is done by T lymphocytes and B lymphocyte. But one thing we need to remember, when a microbe enters into the body, immediately we don't go with with T lymphocytes and B lymphocytes. Third line of defense is not happens immediately. They need to cross the first line and second line. Most of the microbes are going to die in first line and second line. If we fail in first and second line only, we'll come to the third line of defense. What is the basic difference between the first line and second line with the third line? First line of defense and second line of defense are non-specific lines. We don't have any immunological memory with that kind of defense. Then third line of defense, when we interact with the T lymphocyte and B lymphocytes are interacting with the microbes, and when they do the destruction, we get immunological memory with T lymphocytes. Earlier in the previous parts we have seen T memory cells, B memory cells. These memory cells will remember that particular microbe, and when the same microbe enters into the body, they are going to recognize, they kill immediately. That we say immunological memory. The first time when microbe enters, it is a time-taking process to develop the memory. But second time, we are able to kill the microbe immediately because of our immunological memory. That's why very important part of our immune system is third line of defense, which is highly specific but takes several days to become fully functional. But to reach to the third line of defense, a microbe should cross these two lines of defense, first line and second line. Let us start the discussion. First of all, let us see the first line of defense. In case of first line of defense, we already said this is non-specific line but very fast reacting in response. Examples, skin, which is the largest organ of our body, that chiefly act as a physical barrier, means just stop the microbes which are invading germs are going to be stopped by this particular skin. All over the body we have skin, largest organ of body we say, that's why. And it stops the microbe. This we say physical method of stopping, that we call such kind of barrier as physical barrier. And the second one, lysozyme, which is present in the sweat, mucus secreted by the wall of gut, saliva, tears. They're all having a special kind of enzyme called lysozyme, which is more effective on the bacterial cell wall. They dissolve the bacterial cell wall and they do destruct the bacterial cell wall. That lysozyme is present in the sweat, mucus secreted by the wall of gut, saliva and tears. Next, about sebum, which is the low pH in nature. The low pH in nature itself kill the microbes. Sebaceous glands are going to secrete the sebum. And the mucous membranes, the various kinds of mucous membranes, like in the respiratory tract, in the alimentary canal, will have the mucous membranes. Mucous membranes are going to trap the microbes here, and the lysozyme and sebum is considered as a physiological barrier. Earlier we said skin as a physical barrier. Lysozyme and sebum are physiological barrier. Whatever skin, lysozyme, sebum, mucous membranes are all come under first line of defense. Second line of defense is also non-specific line. Like first line of defense, it is also very fast reacting response. Antimicrobial proteins produced by epithelial layers of different parts of the body. Antimicrobial proteins produced by epithelial layers of the different parts of the body is one of the example for second line of defense. And the microbes entering the body through inhaled air are caught in the mucus of the bronchioles and bronchi, and the mucus along with the killed microbes is swept out by the ciliature of the lining epithelium is a process we say second line of defense. Microbes entering the body through inhaled air are caught in the mucus of the bronchioles and bronchi of the respiratory tract, and the mucus along with the killed microbes is swept out by the ciliature of the lining epithelium is considered as second line of defense. And phagocytes, tissue fix and wandering macrophages, like earlier in the previous parts we are seeing histiocytes, Kupffer cells, microglial cells. These are all the different kinds of macrophages in the different parts of the body are also a part of second line of defense. That means without any immunological memory. Then a special kind of cell called natural killer cell, which are considered as granular lymphocytes, is also a part of second line of defense. High temperature, fever. Fever is also one of the immunological technique here. In general, microbes along with our body temperature 37 degrees centigrade, microbe feels so comfortable. So by increasing temperature to 1 or to 2 degrees centigrade, that microbes may kill because of raised temperature. So that's why fever is also a kind of immunological response. And inflammatory responses. Inflammatory responses which is caused by the inflammatory mediators like mast cells and basophils. These are all going to create the inflammatory response, is also a part of second line of defense. So antimicrobial proteins, the mucus along with the ciliature of lining epithelium, phagocytes, natural killer cells, high temperature, inflammatory response are said to be the non-specific second line of defense, which is very fast reacting response. So moving on to the third line of defense, we discussed already about the third line of defense in the previous parts like humoral immunity and cell-mediated immunity are considered as third line of defense, which is highly specific, but it takes several days to become fully functional. Now here a question raises here, how do we develop immunological memory, and what is the difference between a microbe and antigen? What is the difference between a microbe and antigen? Do the microbe and antigen is same? Maybe not all the times. For example, if you take a microbe like this, a kind of microbe is present, we feel so this is not human here, just for our easy recognition I am giving here. These are all the different kinds of microbe. Some 1000 microbes are entered, such kind of 1000 microbes are entered in our body. What we do? Our macrophages will come here. Macrophages. One of the process I am explaining here by taking macrophages as an example. The macrophages engulf these particular microbes. So these are all the different kinds of microbes which were entered in the body. Some 1000 microbes are entered in the body. So when 1000 microbes are entered in the body, the macrophages came here and they are engulfing this particular microbe. Some of them, that means in case of 1000, some 10 microbes are engulfed by this particular. So then these 10 microbes are processed, what I mean, fragmented and cut into different kinds of pieces. So macrophages engulf and they made fragmentation, fragments. We say generally as process. Among this particular processed microbe, one of the processed part is going to be presented on the cell surface. We say that as antigen. Means all the times maybe total microbe cannot be an antigen. A part of microbe, a processed portion of the microbe can be considered as antigen. In general, in case of third line of defense, why we are going to late? Why we are going to respond late here? Imagine what I said now, there are 1000 microbes entered and we are doing with some 10 microbes taken into the macrophages, we process, we fragmented, and macrophages are presenting these particular Macrophages are presented this processed antigen to the T helper cells, and T helper cells will move on to the B lymphocytes, and B lymphocytes, we know that B lymphocytes are having an antibody on the cell surface, recognizes this particular antigenic portion, and they need to prepare a specific model here. That information taken by the B lymphocyte, they differentiate into plasma cells and memory cells. It's a series of events will happen. Macrophages are engulfed the microbe, processed piece, one of the fragment is presented on the surface that we call antigen, and antigen is via T helper cell, it was reached to the B lymphocyte, and the B lymphocyte should differentiate, should activate, and need to produce plasma cells and memory cell, and that plasma cells are going to produce a specific type of antibody under FAB region, which is opt to this particular antigen. This process is going to take some time. In the mean period of time, we said there are 1000 microbes entered, and we are doing experimentalization with only 10 microbes. In the mean process of time, like T helper cell informing to the B lymphocyte, and they are producing plasma cells, and their specific antibody production is going to be late. In mean period of time, the leftover 990 they may multiply and they reach in crores. That's why we get infection for the first time. We are not inefficient, we are late for the first time. In general, the first infection going to happen in most of the people, that because of this particular process, developing the specific antibody against that antigen is going to be little bit late. But we got immunological memory. Imagine the similar kind of microbe entered in the body for the second time, the 1000 microbes are entered. We have memory cells. The memory cells are having an antibody which is specific against, and knowledge of that particular microbe, antigenic fragment of the microbe, and they start immediately producing antibodies here. That antibodies, before they multiply, they are going to be destructed over. So this is a general process. For general information we are giving here, why we are going to be late and why we are active. We are during a second infection. When the infected microbe will come for the second time, we will develop memory cells here. That's why we don't do late like this. Like processed antigen to the helper cell, then to the B lymphocyte differentiation into plasma and memory cell. All this process is going to happen for first time. Second time, immediately memory cells are going to be activated over. So that's why second time or third time or further whatever, that with our immunological memory, we are going to kill that particular microbe immediately, because we have a specific antibodies against that microbe. So generally, that's why we say we are late for the first time. That's why we may get infection. But second time, because of immunological memory and the antibodies, we are not going to be late. Immediately we destruct that particular. That's why we use it to say generally, once we get the disease, we don't get it for second time. Thank you. And in the next part, we'll discuss about innate immunity and acquired immunity. SRIKANTH. For videos and question papers of biology, please visit neetsrikanth.blogspot.com. Thank you.